Hello everyone, welcome to the video for week 7 of the Kombucha lab. Week 7 in lab will be a pretty busy week. It's like week 5 again. So basically every single technique that you have learned so far you'll be doing in lab this week. Your pre-lab this week will also be kind of a review of things that you have already learned. And so, like typically happens, your pre-lab this week will be one part Moodle quiz questions and one part paper pre-lab that you turn in. So the Moodle quiz is going to cover variables and graphing. And so it will give you picture of a graph and ask you, what is the explanatory variable? What is the response variable? What is the result? What is the conclusion? Or it might give you a little story of an experiment and ask you those same types of questions. Or it may ask you about counting and calculating. So that's all the types of questions that could be in your Moodle quiz for this week. In addition to the quiz, you're going to make some graphs in Excel. And the thing that's a little bit different this week is you'll be adding standard deviation error bars. So anytime that you calculate an average, it is best practice to give the standard deviation error bars so that other people will know how spread out the data is, because the average just gives you the average, right? And doesn't tell you if all the values are really close to the average or if all the values are really spread out from the average. So the standard deviation will give you an idea of how spread out the data is. So this week you'll be making graphs and using standard deviation error bars. So you can read about those or watch the video in the lab manual. And then when you're ready to make the graphs, you're going to go to the Moodle page. So here we are in week 7. So here is your pre-lab questions. And then you have the graphing directions. So this is going to tell you about the graphs to make, tells you what data set you should get, what your pollinator is. This will come in handy in a minute. And then it tells you what the 2 graphs are that you should make. So you're each going to make 2 graphs. And it tells you if you just go through the directions. What I would do actually is print out the directions, and so that as you do each step you can cross it off. This will help you to make sure that you get all the directions correct. And so the first graph will look like this. We'll have these values, we'll have these parts. The second graph will look like this. We'll have these values for the explanatory variable. We'll have these parts to the graph. So just follow these directions. You should be fine. Do not hurry. That will make you mess up probably. This example is to remind you of the different parts of the graph in case you don't really remember them. Then once you have your graphs made, here's what you're going to do. So you'll save your Excel file. It's going to be named this. And then you're going to copy the 2 graphs that you have made into a different program where you also can type words. And so it's going to be Word or PowerPoint or something. This where you put the graphs is not important because you save it as a PDF anyway. So move the graphs out of Excel into something where you can type words. And then for each graph, you're going to write the text results that mentions all values of the independent variable. That should totally say explanatory, but it's too late. I'm not changing it now. It's already done. You will not write conclusions. You just need to write results, and then you will save the file that contains your graph and your written results as this name, and then you will go onto Moodle and upload 2 graphs, your Excel graph and your PDF graph. Okay, so that's all of what you're going to do for your pre-lab this week. And then when you go in lab, you'll be doing, again, stuff that you all already know how to do. The very first thing that will happen after the quiz is your result person is going to take observations because you need to get pictures of the jars before you do anything else to them so that the scobies are not wonkadoodle. And so once you take the pictures, then you'll sample out to make the freezer tubes for the TTC, and then you'll sample out to make new kombucha One thing that's different this week is you're going to be taking a pre-fermentation sample. And so you'll make your tea, like black tea, white sugar, control jar, the same as normal. But after the jar is cooled, but before you add the starter, you're going to take a little bit out of the jar and freeze that as your pre-fermentation sample, and then you'll add the starter. So all in here in the lab manual, and then you'll be making dilutions and plating from the jars that you made in week 5. So again, there's quite a bit to do this week, but it's all stuff that you guys have done before, so I'm really confident that it will go smoothly for you.